Hello, my name is Paul Gloth, and this is my journal presentation about the paper entitled Label-Free Microarray Imaging for Direct Detection of DNA Hybridization and Single Nucleotide Mismatches. First, before moving on to the rest of the paper, it's important to know what a microarray is, as it is the focus of this paper. And they are a method of analyzing an unknown DNA sample. This is typically done by taking a target DNA and reverse engineering a complementary strand to that DNA. And through this complementary strand, you can determine through hybridization if the unknown DNA is the target strand. This is done by adding a fluorescent tag to the reverse engineered strand. And once that fluorescent tag is on there, if it binds to the unknown DNA sample, then it can be imaged and you can determine which of the or what is bound in solution. The reason that this paper is significant is because they have developed a method of determining hybridization, and they've made a method of determining hybridization that does not require fluorescent labeling. The first figure depicts the results of their tests to prove detection of hybridization through their imaging method. And in part A of this figure, you can see the initial non-hybridized DNA in solution. And in part B, you can see the difference in density or in mass of the DNA after hybridization has occurred, which is quantified in part C, where only the double mismatch in an 18 mer strand and the two controls, there was no significant mass change after hybridization, showing that they have successfully detected the binding of the complementary strand of DNA to the target strand through multiple different lengths of DNA and through multiple different types of mismatch. And part D quantifies the change of the percent mass change of the different types of strands, where it can be seen that the perfectly matched strands have almost 100% efficacy in hybridization, and the single mismatched strands have above 80, whereas the double mismatched strands and the non-matched at all strands have little to no hybridization. The second figure in this paper is attempting to show the stability of different hybridizations through ionic denaturation kinetics, and it can be seen in part A of this figure, which is a graph of change in mass density over time through different ionic concentrations of salts, that immediately the 18 double mismatch hybridization begins to denature through the weakest solution, and therefore it can be seen as the least stable hybridization, whereas at 0.625 millimolar salts, all hybridizations will denature, and therefore it is difficult to determine different kinetics through ionic denaturation. And part B of this figure is just showing the real-time images taken of the samples as they are going through this process. And the darker the sample, the more denatured the protein within the well. On this slide, figures 3 and 4 are shown. Figure 3 on the left depicts the mass change of 20 mer duplexes over time. In this figure, one can see clear differences between the denaturation of the different duplexes at time point 105, whereas the ionic denaturation, there were significant denaturation changes for all duplexes at the lower concentration solution. And once this graph was generated, the denaturation process was fitted to exponentials in order to determine the difference in the denaturation, which the decay constants for these exponentials are plotted in figure 4 on the right, where one can see that the longer, more well-matched strands are more stable as anticipated. And this also shows that the SPIR method can, in fact, determine stability and binding of these different duplexes. The two main conclusions of this paper are that, one, you no longer have to rely on fluorescent labeling to determine hybridization of DNA, and this is good because hybridization can sometimes have problems in reproducibility, and it can be not totally reliable in its own reaction kinetics. In addition, the second point is that you can now detect single nucleotide polymorphisms without using mass spectrometry, which can involve bulky equipment and can be very time consuming. So this allows larger quantity of people to be able to perform these types of analyses. That's my presentation on this paper. Thank you for listening.